synapse is a junction. It is the junction between two electrically excitable cells where information is passed between them, usually via a chemical messenger. So here we have a synapse between a sensory receptor and a sensory neuron. Notice that there is a gap between these two cells, and that gap must be bridged by a chemical. So this chemical is made by the presynaptic cell and it's sent to the postsynaptic cell. And the type of transmission that occurs here is a chemical transmission and not an electrical transmission. We call this a synapse, and the space between the two cells is called the synaptic gap or the synaptic cleft. Similarly, we could have a synapse between two neurons. The same thing is happening here. There is a presynaptic neuron and a postsynaptic neuron. Finally, we have a neuromuscular junction, which is also a synapse between a neuron and a muscle. In all these cases, a neurotransmitter is the chemical that is involved. There are several types of neurotransmitters, each with a different function. Here is the structure of a typical synapse. Shown in yellow is the presynaptic neuron. Shown in orange is the postsynaptic neuron. This is the synaptic knob, which is the end of the neuron. This is where it ends, the terminal. The action potential is shown here, traveling towards the synaptic terminal. The synaptic knob contains synaptic vesicles. Vesicles are small packages that are carrying something. In this case, they are carrying neurotransmitters. There are calcium channels, voltage-gated calcium channels, that respond to the changes in voltage. The neurotransmitter is released into the synaptic gap. This synaptic gap is also called the synaptic cleft. The neurotransmitter then binds to ligand-gated channels, and these channels are receptors for the neurotransmitter. The neurotransmitter could be either dopamine, adrenaline, glutamate, GABA, etc. The cell that is beyond the synaptic cleft is called the postsynaptic cell. This could either be a neuron or a muscle. The cell that releases the neurotransmitter is called the presynaptic neuron because it is placed before the synaptic cleft. Now let's take a look at the events that take place during synaptic transmission. Here is the synaptic knob. The white circles are the synaptic vesicles, and the red dots represent the neurotransmitter. First, the action potential arrives at the synaptic knob. Once the action potential arrives, it is sensed by voltage-gated calcium channels that are also present in the synaptic knob. These are shown in pink. There is a lot more calcium outside the cell, and so when these channels open, calcium rushes in and causes exocytosis of the synaptic vesicles that release the neurotransmitter into the synaptic cleft. The neurotransmitter then diffuses across the synaptic gap and finds its receptor on the postsynaptic neuron. These are ligand-gated sodium channels, and so when the channels open, sodium rushes in, bringing positive charges into the postsynaptic neuron, thereby depolarizing the postsynaptic neuron. The postsynaptic neuron is now excited. We call this type of local potential an excitatory postsynaptic potential, EPSP for short. Excitatory postsynaptic potential, which means that the postsynaptic neuron is now excited. There are many neurotransmitters that bring about EPSPs. Two important ones are adrenaline and glutamate. The limbic system in our brain is responsible for emotions, memory, learning, and even motivation. One of the most important neurotransmitters in the limbic system is called GABA. GABA stands for gamma aminobutyric acid. We'll just call it GABA. GABA has an inhibitory effect on the postsynaptic neuron. This is important because the amygdala of our limbic system can produce some very strong emotions such as anger, fear, anxiety, and so forth. So GABA helps to calm down these emotions and fine-tune them. It does this by using chloride channels on the postsynaptic neuron. Now let's see how all this happens. We'll begin with the action potential arriving at the synaptic knob, which triggers the opening of voltage-gated calcium channels. Calcium rushes in and causes exocytosis of the synaptic vesicles. GABA is released into the synaptic gap and it binds to its receptor on the postsynaptic neuron. Remember, these postsynaptic receptors are chloride channels, and these channels open and chloride rushes in, bringing negative charges to the postsynaptic neuron. This causes the voltage to drop below the resting membrane potential, resulting in hyperpolarization of the postsynaptic neuron. This causes the neuron to be inhibited, and we call this an inhibitory postsynaptic potential, or IPSP.